Last time we started with a molecule of glucose and followed its oxidation all the way down to carbon dioxide, and the oxygen got reduced to water. So oxidation is the loss of electrons, or in the case of biological oxidation, the loss of electrons and protons, which means the loss of a hydrogen atom. So if we look at the formula for glucose, it gets converted to carbon dioxide. We start with carbons and hydrogens and oxygens. We end up with only carbons and oxygens, so we have lost or removed the hydrogens. Now the hydrogens are not lost, they're not removed and just sitting around waiting to do anything. Those 12 hydrogens that were originally part of glucose were intermediately carried by the coenzyme and then eventually delivered to the oxygen that gets reduced to water. So oxidation is the loss or removal of hydrogen, and reduction is the gain. So as the glucose gets oxidized through those series of reactions of glycolysis, the transition step, and the citric acid cycle, the coenzymes NAD and FAD were capturing or carrying those electrons in the form of hydrogen atoms that will eventually get delivered to the electron transport chain to generate energy for ATP, but also so the hydrogens can join with oxygen and reduce that oxygen to water. The summary formula only shows what happens to the carbons, hydrogens, and oxygens. It does not show us the energy transformations. So I created these tables that, in addition to the actual molecular formulas, tell you what went into each reaction in terms of the carbon, the electron carriers, and the energy. Your book color codes these molecules in this way, so I maintained that color coding. And for the next exam, the pictures of the glycolysis transition step and citric acid cycle will be provided. You will need to be able to look at them and extract this type of information. In glycolysis, we put in one glucose. During the process of glycolysis, that glucose exited glycolysis as 2 molecules of pyruvate. We needed 2 NAD plus electron carriers that in the process get reduced to 2 NADH because they gained hydrogen. When you look back at the formulas, the molecular formulas, you could count the number of hydrogens on glucose versus the number of hydrogens on pyruvate. There will be fewer hydrogens on pyruvate, so glucose gets oxidized to pyruvate, and those hydrogens that were removed from glucose get put onto or reduce NAD plus to generate NADH. In order to start glycolysis, we had to spend a couple of ATP. We also needed some adenosine diphosphate and 2 inorganic phosphates. That's what this abbreviation is. And what comes out of that is these 2 inorganic phosphates plus 2 phosphates from those ATP are put onto ADP to make 4 ATP. Just a net gain of 2 because we spent 2, we made 4, we get 2 as a net gain. That process is substrate level phosphorylation. Phosphorylation is the addition of a phosphate. In this case, we're adding a phosphate to ADP to make ATP. It is called substrate level because the phosphates were stuck onto an organic substrate. The enzyme removed the phosphate from the substrate and added it to ADP to phosphorylate it and make ATP. I left this black because we do have 2 leftover ADP, they get recycled into other energy transformations. Glycolysis is the 2-pyruvate, 2-NADH, and 4-ATP. We have to get the pyruvate into a structural format that is useful to continue, and that was our transition step. Those 2-pyruvate go in. We also need that coenzyme A that contains a sulfhydryl group that makes the end of the pyruvate more reactive. And what comes out of the transition step is 2 carbon dioxides. So we remove 1 carbon from each of the pyruvates in the form of carbon dioxide. And we have 2 acetyl coenzyme A's. The carbon dioxide is waste. It's 2 of the 6 that are going to exit the overall process of cellular respiration. The acetyl is the other 2 carbons from the pyruvate. Each pyruvate has 3 carbons. 1 exits the system as carbon dioxide. The other 2 are each attached to the coenzyme A. This process also requires the electron carrier NAD+. We oxidize the pyruvate to acetyl-CoA. The NAD+ captures those hydrogens to reduce NAD+ to NADH. The citric acid cycle takes those 2 acetyl coenzyme A's, goes through that cycle of oxidations for the carbon, reductions for the electron carriers, and we finish completely oxidizing those carbons that were originally part of glucose. So each acetyl-CoA goes through the citric acid cycle. Once both of them have completed that, we get the remaining 4 carbon dioxides, and the coenzyme A is released to go back to the start of the cycle. During 1 turn of the citric acid cycle, we need 3 NAD+, but we're going to have to go around that cycle twice, so we need 6 NAD+. 1 turn of the cycle requires 1 FAD, so 2 turns of the cycle require 2 of them. The NAD+ again are reduced to NADH because there are 3 places during that cycle where we have oxidations, and the NAD+ captures those electrons. There's a 4th place where there's an additional oxidation, and the FAD is the coenzyme that captures those electrons. There is 1 spot during the cycle where we generate ATP by substrate level phosphorylation. The ADPs and phosphates come in. The energy generated during those oxidation reactions are used to create that ATP energy. When we look at electron transport and chemiosmosis, that flow of the hydrogen ions across the membrane through ATP synthase, that is the chemiosmosis. It is a terrible name. No one asked me when they named it this, because it's not water flowing across the membrane, it is hydrogen ions. The transport of electrons is what creates that hydrogen ion gradient. The flow of the hydrogen ions back across the membrane is the chemiosmosis. If we go back to the start at glycolysis and count up the number of coenzymes that get reduced, we get 10 NADH and 2 FADH2 from the glycolysis, transition, and citric acid cycle. During the release or removal of those hydrogens in electron transport and the pumping of the protons in chemiosmosis, 1 NADH will yield enough energy to get us 3 ATP, and 1 FADH2 creates enough of a gradient to give us 2 ATP. 10 NADH will give us 30 ATP. 2 FADH2 will give us 4. So just electron transport and chemiosmosis gives us 34 ATP. 2 more from the citric acid cycle gets us up to 36. 2 more from glycolysis gets us all the way up to 38. So we can get a total of 38 ATP for one single glucose molecule. That is a pretty darn good energy yield. Glucose only has 24 atoms. We take it through this stepwise process to extract as much cellular energy as possible in that form of ATP. Remember that the free energy change between the reactants and the products doesn't change no matter how we get there. So we could combust glucose with oxygen and we would get the same amount of energy. It just wouldn't be in the form of ATP energy. It would be as heat and light and possibly sound. Going through all of these stages means that rather than losing energy in the form of heat or light or sound, we can reserve that energy in the form of a cellular chemical energy that is useful to a cell. And that is aerobic cellular respiration. Respiration does not mean breathing when we're talking about cellular respiration. When we breathe, we are supporting cellular respiration. We take in oxygen with our breath. That oxygen is carried in our blood to each of our cells. Our cells carry out glycolysis and the transition step and the citric acid cycle and electron transport. All of that combined is what we sort of talk about as our metabolism. We require oxygen, so we do aerobic cellular respiration. Many other types of life do this exactly like we do. All eukaryotes that can do cellular respiration aerobically do it exactly like this. There are prokaryotes that lack a mitochondrion that can also do cellular respiration. They just have to use their plasma membrane as the surface for that electron transport and chemiosmosis. And all the other reactions take place in their cytoplasm. Respiration at the cellular level refers to that electron transport and chemiosmosis. It doesn't refer to breathing because organisms that don't breathe still do cellular respiration. There is also a form of respiration called anaerobic cellular respiration. There is still glycolysis and the transition step and the citric acid cycle, but during electron transport and chemiosmosis, the final electron acceptor is something other than oxygen. A common terminal or final electron acceptor is sulfur. Sulfur is directly below, in the same period, as oxygen in the periodic table. So it has the same number of valence electrons, but it is larger, so it is less stable. In cellular respiration that's aerobic, oxygen is the terminal electron acceptor. If this were anaerobic respiration and sulfur were the final electron acceptor, we would go from glucose to carbon dioxide, but we would go from sulfur to H2S because sulfur gets reduced and we make hydrogen sulfide. We're not going into the details of anaerobic cellular respiration. Fairly specialized organisms, prokaryotes, do that. But it is just another form of metabolism and reinforces this idea that respiration does not require oxygen. Aerobic respiration does. So this is an example of using sulfur as a terminal electron acceptor. There are anaerobic sulfate-reducing bacteria that are in the waters from an eruption of hydrogen sulfide-producing bacteria, causing this green color. Other form of anaerobic without oxygen metabolism that you might be more familiar with is fermentation. Figure 7.14 deals with lactic acid fermentation. It is called that because the lactic acid is the product, the end result of the process. We still start with glucose. We can do this form of fermentation in our muscle cells when they run out of oxygen. So if you exceed your aerobic capacity, you will be forcing your muscle cells to do work anaerobically, and that is much less efficient. We start with the glucose, we still undergo glycolysis to 2-pyruvate, but instead of going to the citric acid cycle and electron transport, we simply convert the pyruvate to lactate. Going from glucose to 2-pyruvate, we have NAD plus gets reduced to NADH. Going from pyruvate to lactate, the NADH gets oxidized to NAD plus. We don't make any ATP in the conversion of pyruvate to lactate. That means that any cell that is relying on lactic acid fermentation as its energy production only gets its ATP during glycolysis in the conversion of glucose to pyruvate. So the purpose of fermentation is not to produce lactate. Lactate is a waste product. That is part of the truth of no pain, no gain. When you exercise really, really hard, well, you could injure yourself and cause pain. But if you're running or doing something very aerobic and you keep going and then your muscles ache, it's not just from the strain, it is from lactic acid building up in those muscles. Our bodies would take that lactate and through a different process convert it back to glucose. But when we're building up that lactate because we're going harder than our aerobic respiration can keep up, we will temporarily do fermentation. So the purpose of fermentation, again, not to produce lactate, but to restore our pool, our cellular back storage of NAD+. Because when we convert pyruvate to lactate, we oxidize the NADH to NAD+, we reduce pyruvate to lactate. We just get the 2 ATP per 1 glucose that happened during glycolysis. There's no electron transport, there's no giant collection of the reduced coenzymes because we have to have the oxidized form of the electron carrier to keep the only process that is making ATP up and running. So other things that we see lactic acid fermentation would be fermented dairy. You ferment lactose. During fermentation, you make lactic acid. So cheeses, yogurt, sour cream, buttermilk, anything that is fermented dairy relies on bacteria doing lactic acid fermentation to produce the acid that curdles the milk. Soy sauce is also a form of lactic acid fermentation. Alcoholic fermentation has the same energy yield and it's done for the same reason. We only get energy during the glycolysis step. And in order to keep glycolysis continuing under anaerobic conditions, we have to restore our pool of NAD+. Glucose to pyruvate is our glycolysis. I haven't shown it, but this is where the ATP is made. We reduce the coenzyme there. From pyruvate to get to alcohol, we have an intermediate step where we create acetaldehyde. That is the process first of removing 2 carbon dioxides and then converting acetaldehyde to ethanol. We reduce acetaldehyde to ethanol, so we oxidize NADH to NAD+ to keep glycolysis going. Again, organisms that carry out alcoholic fermentation do not do so to produce alcohol. Just like for us, ethanol is a toxin. But the reduction of acetaldehyde to ethanol oxidizes NADH to NAD+. That allows the only system that is producing energy to continue. That means that organisms that could survive either way, and there are lots of bacteria that will survive with or without oxygen. Yeast can survive with or without oxygen. If there is oxygen present, they will always do aerobic cellular respiration because of the much greater energy yield, 38 ATP per glucose. Fermentation means 2 ATP per glucose. The few rare prokaryotes that can do anaerobic respiration can get closer to that 38 ATP per glucose, but not many organisms have the enzymes for carrying out respiration in the absence of oxygen. Many more of them have the enzymes for carrying out fermentation in the absence of oxygen. We looked at strictly oxidation of glucose to carbon dioxide for cellular respiration, or anaerobic conversion of that glucose to lactate or to ethanol. This schematic is showing us other things, intermediates, that get derived during the citric acid cycle. We don't just eat glucose. In fact, we almost never eat glucose. The sugar we ingest is usually table sugar, which is sucrose. That's a disaccharide of glucose and fructose. Or we eat high fructose corn syrup, which is fructose, and that is an isomer of glucose. We also extract our energy from proteins and from fats. So how do proteins fit into this cellular respiration, the aerobic cellular respiration? This is showing us where in each of these boxes are the names of amino acids. We get our amino acids when our bodies can break down proteins into their individual amino acid components. Several amino acids can be converted to pyruvate. That's the end of glycolysis. We can break that into acetyl-CoA and go into the citric acid cycle. Isoleucine can be converted directly to acetyl-CoA. Several amino acids to acetoacetate that goes to acetyl-CoA. Some here that get converted to alpha-ketoglutarate, to succinyl-CoA, etc. So we get all 20 of our amino acids can in some way feed somewhere into the overall cellular respiration. In order to do this, first they have to undergo deamination. So amino. We remove the amino end of the amino acid. That means we take the nitrogen off. And now we have hydrocarbons going into, well, with oxygens also, into our organic acid, citric acid cycle. In a very cartoon situation, we saw that some amino acids feed into the citric acid cycle. Some will go into pyruvate oxidation. Fatty acids also enter at this stage. We can break 2 carbons off at a time from the hydrocarbon skeleton of a fatty acid and convert those into acetyl-CoA, and that will continue. And in glycolysis, all carbohydrates will undergo breakdown and rearrangements to feed into glycolysis. Some amino acids can feed into the end of glycolysis, and glycerol, that backbone, the sugar backbone of a triglyceride, glycerol is a 3-carbon sugar, so it can start at that midpoint of glycolysis. Glucose is so important. You've heard of our blood glucose levels. So GLUT4 is a transporter. It's stored in vesicles. So this orange line represents a membrane. The GLUT4 transporter is here. And when we have the presence of insulin, this will fuse with the plasma membrane, and that allows the glucose to go into the cell. So persons who don't make enough insulin, then they don't get this vesicle to fuse, the glucose doesn't go into the cell, and then they have high blood glucose levels because it's not entering into the cells to get metabolized. This is once again the glycolysis pathway with all of the atoms shown. The numbers are showing the different steps, and there are 3 places where there are boxes around the step at step 1, 2, and 7. That is where this process gets regulated. Anytime we're talking about the regulation of a chemical cellular process, we are talking about regulation of the enzyme carrying out that process. The first step is the hexokinase. We regulate the hexokinase enzyme based on the hydrolysis of ATP. So if we have a lot of glucose, we have a lot of substrate for this enzyme. And in the presence of ATP, that enzyme will be active to convert this to the next step. The second enzyme is our phosphoglucose isomerase. We start with a glucose that has phosphate on the number 6 carbon. We end with a fructose that has a phosphate on the number 6 carbon. So when we have a buildup of the glucose 6-phosphate is a signal to the cell and this enzyme to convert this to the fructose 6-phosphate. That changes our sugar from being an aldose, an aldehyde on the carbon number 1, to a ketose, where we have a ketone on carbon number 2. Step number 7 is regulating, getting from our 3-carbon sugar that has 2 phosphates to removing one of those phosphates. Having this molecule in abundance is the signal, and we also have to have high amounts of ADP. If we have high levels of ADP, says we still need to make more cellular energy. We need more ATP. We need this to proceed. So high ADP and a large amount of this molecule is the signal for this enzyme to proceed through the end of glycolysis. Again, this image will appear, so if I ask you what happens at the regulatory steps, this image with the regulatory steps would be circled and indicated for you. A little briefer this time because the metabolism chapters are dense and there's a lot of information. I have some practice items in the module. There's a metabolism worksheet. I will get an embedded video of the ATP synthase that shows specifically and physically how the hydrogen ion gradient is used to synthesize ATP.